So once your PyMINEr analysis has finished running, as you can see it has here, there are a couple different ways that you can go about interpreting and looking at the results and exploring them. The easiest, especially if you've never looked at a PyMINEr results before, is to go over to that example directory here and open up the PyMINEr summary web file. And so here you can see the website that is created, where there are a bunch of different tabs at the top that you can click on, and explanations of what those tabs are. On the home tab, there are first annotation files, where you have TSV of the all of the genes that are in your data set. And if you weren't using human, then the human orthologs will also be gotten. And I'll take you through interpreting the results side by side, both with the website here as well as the other way to look at the results, which is interactively in PyMINEr or in Python. So first we'll open up Python, and then from PyMINEr objs, standing for objects, import PyMINEr object, or PyMINEr obj. All right, so then we are already in the data directory, which is the same as the download/example airway directory over in the other way. And so all you have to do then is PyMINEr obj, and then period, because you're already in the directory. But to be explicit, you just give it the output directory that hosts all of your files. And then we'll make that equal to PyMINEr res, and then it loads in all of the data files. So one of the annoying things about going through someone else's programmatic way of representing, a large complex data structure is that oftentimes you have to go flip back and forth between the documentation in one tab and the actual data structure that you're working with interactively. To get around that, I've tried to embed the help and documentation inside of the files themselves. So if you just print out PyMINEr res, here we have an explanation of what it is. So here at the first level, if you go PyMINEr res dot gene anno and print that, you'll get an explanation of what is all contained within that object. And so here, if we look at that, and then we see that there are two objects of interest there, we can do dot annotations, and then this is just a pandas data frame that has all of the annotations for your genes. And similarly, if we want to look at the human orthologs, again, a pandas data frame describing all of the human ortholog mapping that happened. So that's the results from the gene annotations. Let's go back to the top level and look at the PyMINEr results again here. So we have the PyMINEr clustering results. There's a lot in there. And I'll walk you through that first over on the website. So if we look at the clustering results, there are a whole bunch of images that are produced that you could just pick, some of the prettiest, most informative ones that work for you and throw them into a figure. And there's PCA projection, a heat map of the sample genes, of a sample of the genes that were used for the clustering, a sample of what was actually used for the clustering, the underlying representation, which is actually an affinity matrix. A UMAP projection is another. So for making your figures, you could just pull out a couple of these plots and embed them there. So there's also the TSV file, which has your cell names and which cluster number they belong to, and all of this stuff. You can save externally and then open it up in Excel, and then for people that prefer Excel, it's a lot easier to look at there. So again, if we look at the interactive programming way of looking at those results, we'll print out pmres.clust, and then there's a lot here. So there's actually a little bit more here than in the HTML. So here for sample level info, it's just a vector of your sample IDs. This might be useful for something else that you might want to do with the data set, if you want to integrate some other properties for each of your cells or something like that, or just playing around with the data itself. Sample index dict is a dictionary where you have the sample names, the cell names, and then what index they are. This is essentially what column in the matrix they were for your input matrix. And then labels is just that second column of the of the example I just showed you, where first column is your cell names, second column is the vector of what cluster number that cell belonged to. And then cluster lists is a list of lists that gives you your first list, contains all of the indexes of the cells that belonged to cluster 0. Your second index in that list is a list of indexes that contain the cells that belong to cluster 1, etc., going all the way through. That's more just housekeeping stuff. Marker genes is a little bit more interesting though. So if we go over here and look at the marker genes tab, this first heat map is a heat map that shows, with very strict cutoffs, very exclusive marker genes for each cluster. And the cluster numbers are along here, bottom, and is also colorized up at the top. Now this other second heat map could be potentially more interesting. I find usually gives more usable results, where it's a little bit more flexible with its cutoffs as to what's called a cell type marker gene, where it doesn't have to be truly exclusive, but it can just be exclusive or very enriched in a single cluster. And as you can see here, the heat map is just pulled out from the top 5 best marker genes from each of your clusters, and then concatenating them out like that. And here again, you have the links to the TSV tables that you can open up in Excel. And if we go back over here and we look at this sub-object from the clustering results, we'll take a look at best markers, which was that second heat map, and then all markers. Oop. All right. pm result.clust best markers. So these are just the genes for that second heat map. By default, it just prints out 5. And if you want to take a look around at all of the marker genes, then we'll look at all markers, and this is just a pandas data frame that contains the genes, the symbol, some other annotations. The last column here you have the best group match, but second to last column here, specific marker for group. These are actually the annotations for what was in that first heat map that I showed you over here, where you can see that there are some that actually look pretty good, but not all of them look like they could be super informative. So it's worth playing around if you're looking for marker genes, both at looking at the best marker genes, as well as you can play around with some of the results from this pandas data frame that contains some metrics about how exclusive and how enriched each of these genes is for a given group. All right, what's next in the clustering object? The expression statistics. So I don't have this embedded in the HTML web page of the results, but there are, if you look at this directory, it's sample clustering and summary, expression metrics. It's basically just all of these TSV files that you could open up in Excel. And if you're familiar with some single-cell RNA-seq metrics, these will not sound super foreign to you. So the global non-zero mean is just for each gene, what's the global expression, the average expression of that gene if you remove all of the zeros from that row. And then next is the cluster level mean non-zero expression. So we can take a look at that. And here's the pandas data frame that has the variable name, and then for each sample group, it gives you the non-zero mean expression. Infinity presumably is when it's not expressed at all, actually. But so you'll want to ignore those because that was when the denominator dropped to 0. Let's see, what's next? There's also the ratio non-zero mean expression, which is basically just this file divided by this along the rows, where it's basically saying what's the average expression within a cluster relative to the average non-zero expression globally. You can use that as a metric of enrichment if you like. There's global percent expression, cluster percent expression, and then the ratio of the percentage expressed, again just another metric of looking at how enriched a gene is in a cluster. And then cluster level mean expression, including the zeros and standard deviation. And these are almost all pandas data frames, I believe. And then there's some other expression enrichment, where we have an enrichment table. It has cluster level z-scores, and then a Boolean table that tells you true, false, whether or not a gene is enriched in the given cluster. We can take a look at that. So the enrichment table, this actually has sample level enrichment rather than cluster level enrichment. And then we can look at the Boolean table. And so here we have each sample group, and then true, false if this gene was significantly enriched in that sample group. So what's next? So the significance table is just one-way ANOVAs on whether or not there was differential expression globally amongst any of the clusters. Again, it's represented as a pandas table. And now the pathway analysis is actually fairly interesting. So once you're trying to annotate your clusters, this can be useful. So if we look at pathway importance, here we have basically print out a little bit less. So here we have on the left, we have the importance metric, and this is related to how much information is contained in the manner in which these pathways are differentially enriched across your clusters. So for example, if you were to just sequence whole blood, in almost every pathway analysis that you did on the enriched genes, you would have enrichment for immune system related genes, right? But just the general pathway of immune system doesn't really tell you what's unique and interesting about cluster number 0 from cluster number 1. So what this importance metric is saying how much information is contained within this specific pathway. So we can actually see here the first most significant is actually related to the cilium. So if we just look at the first row, then we'll see here that sample group 3 is really enriched for cilium related gene expression, and then nothing else is. So this group is very clearly the ciliated cells. So you can sort each of these columns, and this might actually be more easily done in Excel. So if you want to do that, you can open up the individual class importance TSV file, which also is over here under cluster pathway analysis, pathway enrichment for each group, and then individual class importance. So we'll save that, and then open it up. So here, again, you can see cilium is the most informative pathway. And if we sort this column, then we get a bunch of extracellular, plasma membrane, redox, ion transport, morphogenesis, etc., and some other developmental pathways. So this might be a stem cell or something like that. Anyway, you can also look at the negative log 10 p-values for all of the pathways that are enriched in each of your clusters. All right, what else do we have in the clustering object? That's actually most of it. And then there's some interesting info in here for if you want to make your own custom plots using the same type of layout of the automated plots. Then there's a bunch of detailed information here. I won't go through all of it just for expedience, but there's a network graph embedding of all of your cells. There's heat map related arguments and annotations, as well as the 2D embeddings for UMAP and PCA, and then just some other useful variables that you can explore with if you want to do your own custom plotting. All right, that's the end of the clustering. So genes of interest, and there's not a lot in the interactive here. It just is the directory and the shortest paths matrix if that was created. In this case it was, because we gave it to the genes of interest to the PyMINEr run. And what this means is, in the network graph representation of the co-expression network of how all the genes are connected and correlated with each other, how far away is every other gene from each of your genes of interest. And if you want to look at your genes of interest here, you'll see some very clear, easy to spot marker genes were our genes of interest here, and they came out in the clusters very cleanly. Here is a bunch of genes related to psyllium, as you can see before. Cluster number 3 is very enriched for those. And so there's a bunch of other plots as well as the heat maps. And this is an overlay of the PCA projection with this gene and all of the other genes for the PCA projections. And then as you continue to scroll down, eventually we'll get to, here's the UMAP, which has, in this case, keratin 5 expression, clearly very dominant over in this area of the UMAP. And then you can, if you're using the genes of interest to help you annotate your clusters, then you can go back over and look at the clustering UMAP here, and then see that it is cluster group number 1 that actually had the highest level of keratin 5 expression, suggesting that this is a population of basal cells for cluster sample group number 1. And then yeah, so that's most of the genes of interest. And then what else do we have after the genes of interest? Next, we have the network, co-expression network. So we have the communities, which has, for every gene, what was the community that it belonged to, what's its local page rank, which is an algorithm I haven't published yet, but I find useful, as well as the global page rank. And if we want to explore that over here, we can look at gene expression networks. So these are what's called the hairball representation of the Spearman co-expression network, and this is what it looks like colorized by the community, where each point here is a gene, and how clustered and close it is to the other genes means that it's more similarly regulated. So it shows a more similar correlated expression pattern. And so if we want to look at individual communities of interest, we can take a look over here. So community 14 has 492 genes in it, and it's very clearly related to the cilia. And it's very highly enriched in cluster number 3, which is exactly what you would expect. And there's also statistics on the differential module use for it. So you can look at the individual genes that are in each of those communities, and then also look at the differential module usage. So does group 0 versus group 1 have more or less, etc. And most of these comparisons are actually significant just because of the number of genes in a cluster, there's a lot of power in those comparisons. So even these subtle little differences often do come out to be significant. And then if we look over here, it's the subset of where that network specifically is in the hairball view. All right, so that is most of the network level analysis. And then there is also z-score overlay plots that show you for each individual gene what is the sample group's level of z-score enrichment. So if you remember, sample group number 1 was a basal cell population that was high for keratin 5. So whatever this module is over here, there was definitely some enrichment there. And sample group 3 was our ciliated cell population, and you can see that community that was very related to cilia is bright and bold and red there. And that about wraps it up for the network analysis. So the last and final thing that's included here is the autocrine and paracrine signaling networks. No fancy graphics for this, unfortunately. For the interactive PyMINEr analysis, it's basically what we just went through, but with more detailed analyses that are preloaded as pandas tables. So if we go over here and look at .ap for autocrine paracrine, there are some objects of interest. So if we look at the first one, this is a table that shows you the number of autocrine paracrine signaling molecule pairs between each of your groups. And that's, fairly self-explanatory. What else did we have available? And then it's pathway analyses, essentially, that were similar, well, basically done in the same way as the pathway analysis that I was showing you before. So there's the pathway negative log 10 p-values. So for each of the cluster pairs, what's the pathway that is signaling most enriched? And these values are, of course, the negative log 10 p-value, and then the importance metric is, like it was in the first time I was explaining, what pathways are giving you the most information. So if we just look at the top results here, extracellular matrix, I guess that's not super surprising. A lot of extracellular matrix sort of stuff, but you can also search for it within these tables pathways of interest. So if you want, you can look at the individual pathways, and then here I'll just open up the negative log 10 p-value in Excel. Open it up. Might take a second. And then if you want to look for something related to developmental signaling, so tissue development, we'll see what is the pair that has the most going on here. So this would be sample group 1 and 5 are signaling quite a lot to each other in this tissue development related pathway. And you'd expect similar results from anatomical structure development pathway, and that is actually exactly what you see. So if you have pathways of interest that you're trying to study, you can use these tables to explore around and see which of the clusters are communicating to each other using what pathways, especially pathways of interest. And then once you have a pathway of interest, you can go over here to a detailed summary of the pathway signaling across and within groups. Oops. It's not loaded in the web file, but save it and open it up in Excel. This file can get very large, just as a warning. So in some cases it might be useful to look at it in the command line if you know how to do that. I'll also open up that -log10 p-value file again, just so I find the where was it? Tissue development. Alright, so we're going to look for this Go pathway and specifically look at the sample group 1 and 5 pairing. So if we search for this, find it here, and sample group 1 and 0. Alright, let's set up a filter. And then should be sample group 1, and then sample group 5. And then we'll search again. Here it is. Alright, so then these are all of the genes that are actually involved in this pathway. So now you can focus in on specific gene candidates and hone in on molecules of interest that you can go after. And then if we look at the detailed summary of all of them, then we can take that gene list that we were just looking at here and then get all of the nitty-gritty details, which include if there's a score which is pulled from string DB, which tells you about the level of confidence that you should have in it, and then if it's directional, is it inhibitory, is it binding, or is there any other information that you can get from it? And to look at it in a little bit more detail, this column is actually fairly useful. So we can open up a new tab and then transpose that. That didn't work. Right. We have to first separate it based on the first level is a comma. So then we'll transpose that. And then we have all of these pairs now, so that going from group 1 was producing CDC42, which has a predicted interaction with FLNA, and all of these other interactions here. Now, you'll probably see some of these that are a little weird, like, for example, CAV2 interacting with EGFR. CAV2, caveolin, of course, is a membrane-associated protein, but it's on the cytosolic side. So the results from this of this interaction of this interaction lookup is actually really only as good as the underlying string db and Go annotations. So there are going to be some that aren't that interesting, but there will be plenty that are definitely real, like EGFR and PDGFRA, etc. That you can look in and help you hone in on your candidate genes of interest that are related to autocrine and paracrine signaling between your clusters of interest or your pathways of interest. All right, and that about wraps it up for the walkthrough of the results, both by the interactive Pythonic way as well as the interactive HTML file summaries. So feel free to get in touch if you have any questions, and look around for some of the extra tutorials that will also cover how to use really large files in HDF5 format, how to convert file formats, and other things like that. All right, thanks for listening. Take care.